Everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimate to you. In the last session, we ended up with key enzymes for gluconeogenesis. In today's session, we will start with significance of gluconeogenesis, in which liver plays a major role in maintaining the blood glucose level. Next, we have substrates for gluconeogenesis, in which lactate, glucogenic amino acids, glycerol, and propionyl CoA play a major role. Then we have regulation of glucogenesis, in which we have pyruvate carboxylase, fructose 1,6 bisphosphatase, ATP, and hormonal regulation of gluconeogenesis. Good morning, everyone. Let us start the topic. Significance of gluconeogenesis. Only liver can replenish blood glucose through gluconeogenesis because glucose 6 phosphatase is present mainly in liver. So, liver plays the major role in maintaining the blood glucose level. During starvation, gluconeogenesis maintains the blood sugar level. The stored glycogen is depleted within first 12 to 18 hours of fasting. On prolonged starvation, the gluconeogenesis is speeded up and protein catabolism provides the substrate, namely glucogenic amino acids. The energy requirement. The reactions catalyzed by pyruvate carboxylase, phosphoenol pyruvate carboxykinase, and phosphoglycerate kinase are required 1 ATP each. So 3 ATPs are used by one pyruvate residue to produce 1/2 molecule of glucose, or 6 ATPs are required to generate one glucose molecule. 2-pyruvate converts to oxaloacetate and 2 ATPs are generated. 2-oxaloacetate is reduced to 2-phosphoenol pyruvate and 2 ATP are generated. 2,3-phosphoglycerate are converted into 2,1,3-bisphosphoglycerate and this also generates 2 ATP. So in total 6 ATP are generated. On the other hand, glucose is converted to lactate and later gluconeogenesis utilizes 6 ATP. Lactate and glucogenic amino acids are the most important substrates for glucose. Lactate formed in the muscle is transported to the liver. In the liver cell, lactate dehydrogenase converts lactate to pyruvate. The pyruvate enters the glucogenic pathway from glucose. Yes, in this diagram, we can see reversal of step 10 of glycolysis. Lactate is converted to pyruvate with the help of enzyme lactate dehydrogenase and conversion of NAD plus to NADH. Second substrate is glucogenic amino acids. Glucogenic amino acids are alanine, glutamic acid, aspartic acid, etc. When glucose is now not readily available, for example, starvation or diabetes mellitus, the glucogenic amino acids are transaminated to corresponding carbon skeletons. These then enter the TCA cycle. Then these then enter the TCA cycle and form oxaloacetate or pyruvate. Alanine released from the muscle is the major substrate for gluconeogenesis. Muscle wastage seen in uncontrolled diabetes mellitus could be explained by this factor. Substrates for glucose. Glucose alanine cycle or Cahill cycle. Alanine is transported to liver, transaminated to pyruvate and then converted to glucose. This glucose may again enter the glycolytic pathway to form pyruvate, which in turn can be transaminated to alanine. Glucose alanine cycle is important condition in starvation. In this diagram, we can see glucose alanine cycle. Lactate and alanine in the liver present are converted to glucose. From here, the glucose enters the muscle cell through blood, and in the muscle cell again, this lactate and alanine are regenerated. These lactate and alanine once again enter into the blood and reaches the liver, and in this way, glucose alanine cycle goes on. Thus, net transfer of amino acids, that is nitrogen, from muscle to liver and corresponding transfer of glucose, that is energy, from liver to muscle is effected. Alanine cycle is intimately related to Cody's cycle. Third substrate is like glycerol. The glycerol part of fat is phosphorylated in the liver cytosol by ATP to glycerol 3 phosphate. It is then oxidized to dihydroxyacetone phosphate by the NAD dependent dehydrogenase. In this diagram, we can see gluconeogenesis from glycerol. Glycerol is converted into glycerol 3 phosphate with the help of enzyme glycerol kinase. Then this glycerol 3 phosphate is converted into dihydroxyacetone phosphate with the help of enzyme glycerol 3 phosphate dehydrogenase. Fourth substrate is propionyl CoA. Propionyl CoA is formed from odd chain fatty acids and carbon skeleton of some amino acids. It is converted to succinyl CoA. It is a minor source for glucose. The important point here is that even chain fatty acids cannot be converted to glucose. They are not substrates for gluconeogenesis. Coming to the next point, regulation of gluconeogenesis. Gluconeogenesis and glycolysis are reciprocally regulated so that one pathway is relatively inactive while the other is active. The regulatory steps are pyruvate carboxylase. It is an allosteric enzyme. Acetyl-CoA is an activator of pyruvate carboxylase. So the generation of oxaloacetate is favored when acetyl-CoA level is sufficiently high. Second regulatory factor is that fructose 1,6 bisphosphatase. Citrate is an activator while fructose 2,6 bisphosphate and AMP are inhibitors of fructose 1,6 bisphosphatase. All these three effectors have an exactly opposite effect on phosphofructokinase. In this diagram, we can see fructose 1,6 bisphosphatase and PFK, that is phosphofructokinase, are reciprocally regulated. Fructose 1,6 bisphosphate has an activator as citrate and inhibitors are AMP and fructose 2,6 phosphate. On the other hand, phosphofructokinase has AMP and F fructose 2,6 bis factor. Fructose 2,6 factor phosphate are activators while ATP and citrate are inhibitors. Third regulatory factor is ATP. Gluconeogenesis is enhanced by ATP. Fourth factor is hormonal regulation of gluconeogenesis. The hormones glucagon and glucocorticoids increase the gluconeogenesis. Glucocorticoids are induce the synthesis of hepatic aminotransferases, thereby increasing gluconeogenesis. In this diagram, we can see hormonal regulation of gluconeogenesis. Glucose and corticoids are activators of gluconeogenesis, while insulin is an inhibitor of gluconeogenesis. On the other hand, insulin is an activator of glycolysis. The increased glucagon to insulin ratio also favors increased gluconeogenic enzymes, which are PEPCK, that is phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. At the same time, synthesis of glycolytic enzymes are hexokinase, phosphofructokinase, and pyruvate kinase are reduced. Insulin inhibits the process. Coming to, coming to GN ratio or DN ratio. During gluconeogenesis, proteins are degraded and end product urea is excreted through urine. The ratio of glucose, that is dextrose, to urea nitrogen in urine is termed as GN ratio or DN ratio. In such animals, the N ratio is 3.65. That means 1 gram of nitrogen from protein will form 3.65 grams of glucose. Since proteins contain 16% nitrogen and 58% is gluconeogenic, GN ratio is enhanced when catabolism is increased. Example are insulin deficiency, starvation, pyrexia, hyperthyroidism, and cancer. Coming to the next point, clinical significance of pyruvate. Pyruvate carboxylase deficiency. It is seen in inborn error of metabolism, where mental retardation is manifested. Its incidence is 1 in 25,000 births. Lactic acidosis is noticed. Second point is malignant hyperthermia. This may occur when halothane is given as an anesthetic to certain persons. The rhizonidine receptor, a calcium release channel, is defective, leading to inappropriate release of calcium from sarcoplasmic reticulum. This results in uncontrolled heat generation, damage of muscle cells, ATP depletion, lactic acidosis, and rhabdomyolysis. CPK, that is creatine phosphokinase, is markedly elevated. This defect is seen in 1 per 50,000 population. Ethanol or ethyl alcohol. It inhibits the gluconeogenesis. During the metabolism of ethanol, the level of cytoplasmic NADH is increased. Thus, the pyruvate to malate to oxaloacetate reactions are reversed. So, excessive ingestion of alcohol results in hypoglycemia. Lactate also accumulates as NADH level is increased. Coming to the next metabolism, the glycogen metabolism. Glycogen is a homopolysaccharide with the glucose units linked in alpha 1,4 linkages, that is straight line, and alpha 1,6 linkages, that is a branching point. Branching makes the molecule more globular and less space consuming. In this diagram, we can see branches are of glycogen which are excessive because of alpha 1,6 linkages, and in the center the circular part is glycogenin protein. Here ends the session. Kindly like, share, and subscribe the channel. Thank you.